All right, protein synthesis. As you can guess, synthesis to make. So now we're going to make proteins. All right, George Beadle and Edward Tatum, two very important men back in 1934, thus the black and white pictures. All right, so Beadle and Tatum, they developed the one gene, one enzyme hypothesis. This proposes that a single gene has the genetic information for making one enzyme. Later, that was changed and became the one gene, one polypeptide or protein. So one gene, one protein hypothesis. So as enzymes are a type of polypeptide, it's a type of protein. All right, so we have two processes that take place here. We have transcription and translation in both prokaryotic and eukaryotic cells. And so obviously a little more complex there in the eukaryotic cells, but it's occurring in both nonetheless. All right, so with transcription and translation, transcription means the process of making a working copy of an original. So you're transcribing from an original copy. So the process is in the making of a recyclable, workable copy of DNA, but it's got to be in the form of RNA. So this actual recyclable copy will become known as messenger RNA or mRNA, and it is a recyclable copy of the $1 million DNA blueprint. So let's see, does it show it? No, this goes down to here. Okay, so back to here, transcription and translation. All right, so with transcription, I like to use the example. A big part of what I did while I was in the military is I was in communications. And so what would happen is I would sit in my equipment rig, and someone would bring me a message, and just a regular old message that needed to be sent. So I would type that message, and as I would into a piece of equipment, and as I typed that message, it would transcribe what someone else said into a code. All right, so it would make a copy of the original, and then you're going to see what's going to happen on the other end. So here's this transcription. We're in the DNA. We can't leave the DNA. We can't leave the nucleus. Did I say we're in the DNA? Whoo, it's getting late. We're in the nucleus. The DNA can't leave. The DNA is the $1 million DNA blueprint. It can't leave. If it leaves, we're all in trouble. So there's pores in the nuclear envelope. So in the membrane around it, there's these pores in there. You can see these little holes. DNA is too big to fit through, but RNA is single-stranded. DNA has two strands. RNA is single-stranded, and RNA can leave. So can't let the DNA leave. It's too dangerous for it to leave. And so we have to make messenger RNA. There's three types of RNA. Messenger is the first one, and it's going to do just what it says. It's going to be a messenger. So it's going to make a working copy of the original. Now this is synthesized through an enzyme called RNA polymerase. Surprisingly, huh? So polymerase, many, ACE, of course, the enzyme. So we're making RNA. All right. Now the messenger RNA is going to be sent to what's called the construction site, which is the ribosome. So the messenger RNA is going to leave the nucleus through the pore and go out to the construction site, the ribosome, which is the site of, you got it, protein synthesis. Oh, the story's getting even better as we go along. All right, now, so the RNA nucleotides use the sugar ribose instead of deoxyribose, as is 5-carbon sugar. It's important to note that. So ribose instead of deoxyribose. In RNA, uracil replaces thymine. So instead of ATGC, it's AUGC. So thymine can't get out of the nuclear pores either. So remember, the ribosomes are out in the cytoplasm, so thymine needs to be substituted by uracil. The other thing about messenger RNA is it's single-stranded. There's only one strand. So it's less stable than DNA. DNA is stronger. All right, so the DNA serves as the template. So we've heard that before. So this is going to be the guide for making messenger RNA. We're going to do the same process. In our next video, we're going to go into more detail about opening everything up and making it. But just know the basics here. Serves as the template. Again, adenine pairs with uracil and cytosine pairs with guanine. Still using Chargaff's rule. Transcription is considered the first part of protein synthesis. All right? Now we need to move. So here's transcription that occurs in the nucleus. And we're out here. Now we need to translate it. So here's where I have typed the message, and now it's a coded message and it gets sent out. When it's received at the other end, it's received by someone, but it means nothing until it's translated, because right now it's just in code. And so then someone on the other end has to translate what it is. So the translation means the process of taking from one language and changing it into another. So a good example would be adios in Spanish going to goodbye in English, or hola to mean hello. So what we're doing is, in the process, the cell is turning the nucleotide language, the DNA, RNA, into amino acid language. So we're going from nucleotide language to amino acid language to make proteins. Remember, amino acids make proteins. They are the building blocks of proteins. So the process occurs at the ribosome. This is where that building site is. The ribosome has a nickname called the translator. Dun, dun, dun. You could just hear the music. It's also considered a construction site, since the cell is building a protein using this copied blueprint from the DNA. Translation is considered the second part of protein synthesis. So transcription is the first part, translation is the second part. All right, so here is here's the deoxyribose sugar, and here's the ribose sugar here. And so you can see when it's called deoxy, that means, like, if you're de-icing something, you're removing the ice. Well, notice all of this and this are the same except for the number 2' carbon right here. The number 2 carbon has a hydroxyl group, and this just has that oxygen's been removed, thus the word deoxy. So that's the only difference there. That number 2 carbon does not have the oxygen, the other oxygen hanging off. And of course, then we have uracil instead of thymine. All right, so transcription and translation. When you look here, here's transcription. Here's our original DNA template. The DNA has been opened up. Here's our single strand, and then notice the numbers 5' to 3', 5' to 3'. They're still numbering them. And now we're going to make messenger RNA. And then we translate. We use these triplet code, a codon. This right here, this word codon, is a code. And we call it a triplet because there's 3 letters that will code for a particular amino acid. And then these 3 letters will code for a different amino acid. So the order of these is very important. So you can see one mistake can make a huge difference. So these 2 codons must be read from the 5' to the 3' end on the messenger RNA. This is how the messenger RNA was made. So you don't write a sentence and then read it backwards, do you? It wouldn't make any sense. All right. Now, Nuremberg is kind of like the decoder. He was the one that figured out about the translation. All right, now this RNA codon chart, we use this chart in order to determine what the amino acids are. And so it uses the nucleotide sequence on the messenger RNA, that triplet. So notice there's a first base, second base, and the third base. So when we have something of, say, if we had a codon that was AAU, we'd look here, A, and then A, and then, so if it was this A, then we know it's somewhere in this row. If it's in AAU, then here's the second A, so it's got to be somewhere here where those two cross over each other, and then U is the last one, so we know this is going to be our amino acid. So most amino acids will have more than one codon. There's 20 known amino acids, and AUG is the start codon. UAA, UAG, and UGA are the stop codons, and they'll stop the process of transcription. This is basically uniform or universal for all living organisms. Now, keep in mind, and it's also universal for viruses too, but keep in mind that viruses are not considered living. All right, so we're going to practice some of these. We'll practice actually a bunch of them, but these become really fun. That's all I have. Oh my goodness. Part 1 of protein synthesis comes to an end. Good night.